So GSDMD inhibition is what you are proposing or looking at as a very interesting intervention because it will stop or annul the release of IL-1 beta into the extracellular milieu, right?
Right. I mean, I didn't have time to go into it, but a lot of our work has to do with the cytokine storm because when you get this GSDMD pores and you get this burst of cytokine release, like in COVID, at the end of the day, what kills you in COVID with SARS-CoV-2 is not so much the virus, but it's your response to the virus, which is this cytokine storm. And the real hope of drugs that, if they come to place, to come to fruition, drugs like Gesdermin D inhibitor, is that they would be used in such a scenario where you want to really dampen that storm, push down the curve of cytokine release. So that's that. And we know this from our knockout studies, where we don't have a drug, but we can use the knockout. It's not ideal, but still. So there's a lot of interest in the pharmaceutical industry in developing Gesdermin D inhibitors to blunt the cytokine storm.
Uh-huh. Yeah, I was coming to that. In the contemporary world, you cannot think beyond. Everything sort of brings COVID into the picture, and especially when you're talking about cytokine storm and just D, it looked like a very nice intervention to stop or look at how that annuls the cytokine storm, especially in COVID-like situations. Thank you. There's one last point that I had in mind. How does the cytotoxic T cells play out in the extracellular milieu in the presence of when IL-1 beta is released? I can see that the macrophage will be intervened. The macrophage interaction with, especially in a solid tumor or a cancer scenario, you will have macrophages which will be IL-1 beta induced macrophage maturation to the M2 macrophage, right? How does the cytotoxic T cells play out in this? The CD8 T cells,
I think that there is some literature to suggest a role for the inflammasome in T cells, but it's lesser. So remember, the cytotoxic T cell is not using the inflammasome to kill the tumor cell. It is using granzyme B and Fas ligand to activate the apoptotic pathway in the tumor cell. This is the caspase 8 pathway. So it doesn't have anything really to do with IL-1 beta. I mean, IL-1 beta is a very important cytokine in immunology. It's a priming cytokine, but the cytotoxic T cell response per se has nothing to do with IL-1 beta. That has to do more with the TCR recognition of the peptide in the MHC groove of the tumor cell. Once that recognition happens, that is the trigger for the cytotoxic T cell degranulation.
So what I'm understanding is this could be independent parallel pathways in the context, right?
Yeah. So there's apoptosis, which is a form of cell death, which is really a silent form of cell death because the cell membranes are kept intact in apoptotic demise and they're engulfed by neighbor cells. So you don't get the spilling out of cytokines. You don't get the spilling out. It's usually a silent death. So whereas necrosis is a death that is more inflammatory because you're getting rupture of the membrane. So really two different forms of death.
Thank you so much. Very aptly, so many ways to kill, right? such a beautiful title. Thank you, Dr. Dixit. It was a marvelous time that I had listening to your talk. Thank you so much.
Yeah, thank you, Shantanu, for the comments and the questions. So, Dr. Dixit, there's one question by Dr. Anshu Bharadwaj. What is the physiological role of P30? In absence of infection, how is it regulated?
So, you're not going to get P30 unless you activate the inflammasome, and you're not going to activate the inflammasome unless you have an infection. So if you have a knockout of Gesdermin D, GSDMD, so you can't generate P30, those mice in a clean animal facility will live to old age. It's only when you challenge them with gram-negative or LPS that you see a phenotype. Yeah? So a large part of our genome, and this is true across life, a large part of the genome of bacteria is devoted to fighting off phage. Similarly, a large part of our genome is devoted to fighting off pathogens, so that many of the major players in immunity, you can remove them, but they don't have a physiological function. It isn't like you knocked out phosphofructokinase. But obviously these animals wouldn't survive in the wild because they wouldn't be able to withstand infectious pressures. But that's an important realization. It of course works to our advantage because we can now make these engineered animals and then subject them to controlled infections.
Yeah. So one last question. You mentioned about the therapeutic options for COVID, like the cytokine storm and Gasdermin. So do we know any repurposed drugs which influence this pathway? Because that is something which I think world over everybody is looking for drugs for COVID, and that's an area where we really haven't made much progress. If you look at remdesivir or favipiravir, those interfering with the viral replication. So I think it's a host target which we need to do it, and this would be a good therapeutic target.
It would, I think it's not really my area of expertise, repurpose drugs for COVID, but, there's a lot happening on that front. I'm not aware of any clear winner, that has come to the fore. There's all anti-IL-6 is effective, but these are all, at the margins. We don't really have a good antiviral, repurposed or otherwise.
Yeah. So are there any questions from the CSIR team who is on the panel here from CDRI or anybody else would like to?
Okay. And IL-1 response between the mouse cell and the human cell varies considerably, right? Like if we take the mouse macrophage and if we take a human macrophage or monocyte, then the IL-1 response, the production or the stimulus, they vary considerably in between the mouse and human. So shouldn't he if given that the the effect of these pathways are very essential, shouldn't the response of the animal and human cell be similar, like kind of the IL-1 production?
Yeah, no, it's the production of IL-1 beta is quite similar. But what is really, and I think this is a more general point I'd make, is that when you use animal models, it's almost going to be impossible to model the disease itself. You can model aspects of the disease. So I'll just give you an example that if you look at LPS, that primates are 1,000 times more sensitive to LPS than rodents. So clearly you can't fall into the trap of using LPS in a mouse and thinking that's the same as gram-negative endotoxic shock in primates, right? So I think you have to be very careful. And so we actually use animal models to test our biochemical hypotheses, like you saw that with the Gesdermin D knock-in mouse that cannot be processed by caspase-11, right? We have a hypothesis and we use that. And so we answer a question. But I'm very wary of extrapolating animal models as replicas of human disease, because they just aren't.
Thank you.
Okay, I think there's one last question and we can stop this public lecture with that. There is a school of thought that mitochondria are potential bacterial reminiscent. Are there any mitochondrial components or state that can trigger P30 formation?
I'm not aware of any, and that's not to say some may not come up, but I would actually say the reverse. That I think once you generate P30, you severely damage mitochondrial membranes. And that one reason why this pyroptotic Ges-dermin-D mediated, P30 mediated death is so inflammatory is that P30 willy-nilly, indiscriminately permeabilizes ER, Golgi, mitochondrial, plasma membrane. So it's just really incredibly damaging to the organelle systems in the cell. So I think it's spewing out of those contents, acute spewing out of those contents that contributes to the inflammation.
Okay.
Remember, you have to, just for the students, you have to remember that when you talk about cell death and inflammation, you're talking about a very fundamental biology, because this is the way we regenerate, right? When you have an injury, let us say a blunt injury, sterile inflammation, you run into a table and you develop a big bruise. That bruise is because cells died, they released inflammatory mediators, they recruited cells of the immune system to have a reparative process that allowed for healing. So these things are very highly conserved and they have a very important purpose in our day-to-day existence. So by that I mean cell death induced inflammation. So you really want to think of these things in that more general manner.
Yeah. Thank you very much. I think that's an excellent way to end the talk. When you talk, how do you induce the cell death and how do you even stop it? Because, you don't want it to go beyond the bruise. So it's a delicate balance, right, over there. Like, when do you switch it on and when do you switch it off? And as you said, this is a ninja which can do everything and, kill. So you have to regulate it. And I think that's where the whole balance is of cell death and cell proliferation. And thank you so much, Dr. Dixit, for giving us this wonderful talk. And I hope our viewers who joined us through various social media channels also have enjoyed it. And with this, I think we will stop the public lecture and then we can have our internal discussion. Thank you so much, everybody who's joined us. And Deepak, you can switch off the live streaming now.